Well, first of all, I would like to thank the committee for this incredible award. I was very excited to have been nominated and also to have been selected for this award. So the project I would like to show you today focuses on the development of techniques to spy on viruses. And specifically, I want to spy on RNA viruses. So these are viruses that have an RNA genome. And here I'm just listing some of the very well-known pathogenic viruses. And they range from all kinds of viruses causing all kinds of diseases. So for example, there's the relatively benign common cold causing rhinovirus, but then there are also some very severe pathogens that can cause a very big impact on our health and society, and they may even cause pandemics. And all of those viruses have in common that a typical infection starts with a single viral particle infecting a single cell, resulting in the release of one copy of the viral genome. And then the main thing that the virus wants to do is to replicate. It wants to multiply its genome, produce new proteins so that it can form more viral particles. So along the timeline of an infection, at the end of an infection or later in infection, we will see a cell that has many RNA molecules, many viral genome molecules. Luckily for us, our host cells have all kinds of mechanisms to detect the virus and try to prevent the virus from replicating. So somewhere along this timeline, the host cell may start to engage in this competition between the virus and the host. And as a result, the outcome of a virus infection depends on this complex competition between the virus that wants to replicate and the host cell that wants to prevent it. Actually, it has been quite challenging to study this competition, and that's because most assays do not have the sensitivity to detect those early stages. They do not have single molecule resolution, so they cannot see when there's only one or a few viral RNA molecules. Furthermore, most assays require you to fix or lyse your cells. So that means we can no longer follow over time what's going to happen with that infection because we just killed the cells. And finally, many assays require you to pull a lot of cells at once, but we don't know whether all cells are going to get infected, whether the infection takes place at the same moment, and whether the infection is progressing at the same rate. And therefore, it has been very challenging to study the timing, regulation, and success rate of an infection, and also to really disentangle this competition between the virus and the host. So to overcome that, we decided to develop live cell single molecule imaging assays. And for that, we teamed up with the lab of Frank van Kuppenveld at the Utrecht University to study Picornaviruses. Picornaviruses are small non-enveloped RNA viruses, and their genome is a positive sensed single RNA molecule, meaning that there is only one RNA molecule, and this is directly coding for the viral proteins. The first step is the release of the viral genome. This is this positive RNA molecule, which can then be translated by the ribosomes of the host cell, resulting in the production of a polyprotein. And this polyprotein is then being processed into 10 or 12 viral proteins. And among those, we have the viral polymerase and the capsids. And the RNA-dependent RNA polymerase at one point may start to use this genome as a template for replication. So it first generates a negative sensed RNA molecule, and then this negative sensed RNA molecule is then used as a template for more positive sensed RNA molecules. And those positive sensed RNA molecules can then all be translated, produce new viral proteins, and some of those RNA molecules may serve again as a template for replication. And then ultimately those molecules assemble into new viral particles, the cell lyse, and there is the release of new viral particles. So to enable single molecule imaging, we use the SUNTEC technology. So the SUNTEC technology is based on introducing 5 SUNTEC peptides at the end terminus of the viral polyprotein. And in this case, we're using the Picornavirus CVB3, Coxsackie-virus B3. And then we generate virus with this and we infect stab cells. And stab cells are cells that stably express a GFP-coupled intrabody that recognizes the SUNTEC epitopes. So what's going to happen in those cells is that the viral RNA molecules are going to be translated by the host cell ribosomes, and then those SUNTEC peptides are going to be generated, and they can be rapidly bound by the stab, by the SUNTEC antibody. And in this way, we recruit GFP fluorescence to the viral RNA molecule. Well, a single RNA molecule may be translated simultaneously by many ribosomes, and in this way, we recruit a lot of GFP to our viral RNA molecule of interest. And then we can see it under the microscope as a bright spot. And that's indeed what we can see. So this is a stab cell. We can see those nice GFP foci. And indeed, if we combine it then with single molecule FISH, we can see that those foci overlap with the individual viral RNA molecules. So each GFP spot is a single viral RNA molecule. And now we have an assay in which we can study various aspects related to the virus infection. So in this assay, the brightness of the spot depends on the number of ribosomes and therefore reflects the translation efficiency of the virus. And the number of spots depends on the number of viral RNA molecules and can thus be used to measure replication. And I want to show some data on the replication. So this is a stab cell, it's currently not infected. And now the really nice thing is that we can do this assay live. So the infection will start in the bottom part of the cell. And there we can see there is a single spot. And this single translating viral RNA molecule is the RNA molecule from the viral particle. So this is the genome of the infecting virus. And then over time, we can see that this virus is replicating because there's an accumulation of spots. And once again, each spot is a single viral RNA molecule. And just within a couple of hours, the entire cell is full of new RNA molecules. Well, we did this for many cells. So for example, in these 3 cells, we measured how the infection is progressing over time. And I hope you will agree with me that even though the exact timing, number of molecules, and the duration differs between those cells, they have a lot in common. And that's what we decided to call virus infection phases. So the infections start during phase 1. And during phase 1, we see one single molecule, and this is the translation of the incoming viral RNA molecule. And then during phase 2, there is a period that we don't see any signal. And that's because the RNA molecule is being replicated. Our assay depends on the active translation, so we don't see the replication itself, but we can see the product of replication because we see the translation of the newly synthesized RNA molecules during phase 3, when there is a rapid increase in the number of foci. For reasons we currently don't understand, this increase stops at a certain point during phase 4, and we think that one of those newly synthesized RNA molecules now serves as a template to again replicate, which we then see in phase 5 by more increase in spots. And using this assay, we can now measure the dynamics of replication by, for example, measuring the duration of each phases. So in this graph, we see phase 1, 2, 3, 4, and we plot the distribution of the durations by a Kaplan-Meier curve. So that means all the cells are aligned at the start of a phase, and then if a cell is leaving that phase and starting the next one, there's going to be a drop in the graph. So for example, we see that the duration of phase 3 and phase 4 have a very narrow distribution and take about 30 minutes. From this analysis, we can learn a couple of very interesting things. So for example, if we look at phase 1, we see that about an hour into the infection, phase 1 will have been completed in 50% of the cells. But there are also those cells in which it takes 4, 6, or even 8 hours to complete this first phase, in which it takes hours to complete the translation of the first RNA molecule. As a result of that, if we combine all the cells in an assay altogether and we look at the distribution of how many cells are in which phase, we can see that basically at any given time point, there are cells present in any given phase. And if we now compare this to traditional virology assays, they typically do not get enough signal above background at this 4-hour time point, whereas we can see that cells in any of those replication phases are present. And another thing that we can see from this analysis has to do with phase 2. So phase 2 is the replication of the incoming RNA molecule. And phase 2 takes place very rapidly in 20 to 30 minutes in 80% of the cells. However, in about 15 to 20% of the cells, we see no replication. So for example, in this cell, we can see it's infected. There's the translation of the first RNA molecule. Translation is being turned off. The virus turned off may try to replicate, but it never manages to produce any new RNA molecules. And we only see this for phase 2. So phase 2, the replication of the first RNA molecule is a bottleneck for successful replication. Well, I mentioned that we wanted to use this assay to study the interplay with the host cell. So to do that, I need to briefly introduce the interferons. If a cell is infected by a virus and it recognizes the virus, it may start to produce interferon and secrete this. And among other things, this will result in an activated antiviral state in the surrounding cells. If those surrounding cells would now get infected as well, they are better capable of fighting off the infection. So we wondered, what does that mean, fighting off an infection? So basically, how does an activated antiviral state prevent virus replication? So to do that, we used our assay and then looked at each of the phases. So the data here in black is the untreated control, the data in red is the interferon-treated cells. And then we saw a very strong effect on phase 2. So if we have a cell with an activated antiviral state, we see a very strong repression of replication specifically during phase 2. But we don't see this for any of the other phases. So that means, once again, phase 2 is the vulnerable moment in the viral life cycle, but it's also kind of the only moment in the viral life cycle, because if those cells manage to complete phase 2, they are seemingly uninfected in later phases. So to finish, I've showed you that we have developed a live cell single molecule imaging assay to study virus translation and replication. And using this on Picornaviruses, we've seen that there are those 5 phases. We see that specifically translation and replication of the first RNA molecule is extremely heterogeneous, and that the replication of the first RNA molecule is a potent target for the antiviral response. And this is just on the Picornaviruses, but we think our approaches may be broadly applicable to many of the other RNA viruses that I mentioned on the first slide. With that, I would like to thank all the people involved in this project, specifically the other members of Team Virus in the Tanenbaum lab. This is the Tanenbaum lab in the 1 month that we did not have any lockdown. I mentioned that this is a collaborative effort with the Van Kuppenveld lab, and I also would like to acknowledge Hans and Joep for helping us to expand the assay to intestinal cells. And with that, I'm very happy to take any questions.